Let's go over transcriptional control and the lac operon. So organisms don't need to constantly transcribe every region of their genome, so there are certain controls in place that will help regulate this process. These are helped by activator proteins, which will bind to promoters and upregulate transcription. These are used in positive regulation controls, and repressor proteins, which will bind to the operator region past the promoter region and inhibit transcription, which are negative controls. Control methods will differ between eukaryotes and prokaryotes. In eukaryotes, it's going to be more sophisticated because there will be presence of several transcription factors, and there will also be temporal and spatial regulations put in place because of the nuclear envelope. Because transcription will occur within the nucleus and translation will occur outside of the nucleus, there will be a time, or there will be a separation in space where both occur, and there will also be a separation in time because the mRNA still has to be transported out before being translated. In prokaryotes, because the transcription and translation occurs in the same medium, they can be coupled together. When a DNA is being transcribed into mRNA strands, the mRNA strands can be translated while it's being transcribed, and as such it's a coupled manner of, and so transcription and translation will be coupled. This allows prokaryotes to better adapt to meet environmental needs in a faster method, and these will only use activators, repressors, and in some cases enhancers, but that's going to be rare. Operons can be categorized into two categories. There's inducible and repressible ones. Inducible ones are naturally off but are turned on by metabolites, and repressible ones are usually turned on but can be turned off through metabolites or regulation. Like operons can be categorized as inducible because the metabolite lactose turns it on from its off state, and tryptophan, which is usually on, is repressible because the presence of tryptophan will turn it off. To our right, we have the two different types of regulation methods. We have the positive and the negative. The positive enables the expression of genes, and this occurs when the transcription factors binds to the promoter region and enables RNA polymerase to bind and read the DNA. For this to happen, allosteric effectors or inducers would often have to bind to the transcription factors themselves, and when the transcription factors and the effectors are put into play, this will help enable transcription and turn genes on. As for negative regulation, this occurs when gene expression is prevented. This is caused by protein repressors, which can bind to the operator regions of the gene, thereby blocking the binding site or blocking the transcription of RNA polymerase on the promoter region. Allosteric effectors, or inducers in this case, will bind to the repressors and keep it from binding from the DNA itself, so when the repressors cannot bind, it cannot block the path. If the repressors bind, and if negative regulation is enacted, then this will disable transcription, thereby turning off gene expression. The lac operon, as stated prior, was an inducible system, but it has two different mechanisms in play. We have the repressor protein, which is negative gene regulation, and the cap protein, which is the positive gene regulation. Lactose is going to be one of the effectors, or the inducers, and they're going to bind with the repressor protein whenever they are available. That's why the repressor is bound with the lactose unavailable over here, and the lactose available over here. However, if there is the CAMP-CAP complex with CAMP produced from conditions with little glucose, then it's going to bind over to the cap binding site, the complex, and it's going to allow RNA polymerase to bind in the promoter region and the operating region over here. As we can see, if we put the two together, the glucose and lactose available will unbind the repressor protein and better bind the RNA polymerase to the DNA strand, thereby making the gene strongly expressed. But if there is high glucose, then the complex won't be created, thereby lowering the RNA polymerase binding rate and giving a low basal rate of gene expression. If lactose is unavailable, then the repressor protein remains unbound and it's going to bind to the DNA protein over here, or the DNA molecule over here. In this case, if there is high glucose or low glucose, then there won't be any expression of the gene itself. There are going to be two genes, or two alleles we'll be looking over today. There are going to be the I gene and the O gene. LAC-I is going to code for the repressor protein over here. It's going to be a regulatory gene, and it's going to be located over here. The wild type, the plus type, is going to be able to rescue any alleles which has a negative one over here. And as such, it's dominant to this one. And it enables normal functioning, which is found in this diagram. If it's I negative, then the repressor will be unable to bind to the operator region, even if it's unbound to lactose. As such, the repressor protein still remains unbound, even if lactose isn't available. And it's going to express the LAC genes over here. And then as for the IS to superrepressor mutation, it cannot bind to lactose, and as such, the repressor protein will be able to bind to DNA even if lactose is available. This turns transcription off even if lactose is available, thereby making it deviate from this diagram. So the repressor protein will still be bound in these two cases, and no genes will be expressed. For LAC-O, this will be encoding for the operator region, which is where the repressor protein is bound to. And if we have the OC, then that means the operator is unable to bind to the repressor protein, meaning that no matter if there is lactose or not lactose, then transcription will still remain on, because the repressor protein cannot block the path. This notation will be used for the addition of plasmids of DNA. If there are different alleles for the LAC-I gene, then the plasmid can overtake the effects of the organism's original alleles, because the protein product can still diffuse and either block or keep the normal phenotype over here. LAC-O genes of the plasmids, however, have no effect, because the genes will be found on the track itself, and it can be diffused into the genome of the organism over here. In eukaryotic control, they don't have any operons, and the genes will be on separate strands of mRNA, but they'll still be able to regulate which genes get transcribed when. This is done through two methods: transcription factors. They can respond to changes in the environment and chemicals in the environment, and they will help change transcription behaviors of certain genes, and chromatin modifications, which is over here. Whenever this thing is modified, it's either raveled or unraveled, and if it is unraveled, then certain areas are looser, and polymerase will be able to bind easier and transcribe the region, thereby upregulating expression. To clarify it a bit more, let's go into certain examples. In acetylation, the positive charge of the octameres are neutralized. This will relax the tight winding of the DNA and the histones, and this will allow for transcription to activate, because polymerases can bind better to the looser strands of DNA. Deacetylation is the reverse, where the histone and DNA becomes more tightly packed, the covalent interactions are stronger, and the polymerases will have a harder time getting through, thereby discouraging transcription. Methylation is the addition of methyl groups to these histones over here, and it's thought to be a gene silencing action. And phosphorylation will give a negative charge on the histones, resulting in a more open presentation and allowing polymerases to bind better. This can also be used in DNA damage repair and chromatin remodeling by SWI/SNFs. Okay, and I'll give you a second to answer this. And yes, These are repressors. The LAC-I protein goes over here and binds to the operator region, which is where repressors or inhibitors often bind to. And the effector molecule over here, or the inducer, will bind and discourage binding of this protein, thereby allowing transcription. This is negative control of transcription.